This is Blood and Cancer, the official podcast of MD Edge Hematology/Oncology, where we discuss the latest research, treatment considerations, and other issues related to hematology and oncology. Welcome to this podcast. I'm your host, Dr. David Henry, and this week I'm delighted to be speaking with Dr. Leslie Kean, who's Director of Pediatric Stem Cell Transplant, Boston Children's Hospital at the Dana-Farber Cancer Institute. Leslie, thanks for joining us today.
Great to be here.
So very nice to have you. And Leslie was a co-chair of what I think is one of the best parts of ASH or ASCO is the best of. And she was tasked with doing, as I told her, I'll do the best I can to pronounce these products. So the CAR T abstracts which came out in the session she chaired for the best of. So what we'll do is there's 4 abstracts I'll list, and I'll try and say the titles, and then Leslie will tell us what she thought was the take home and what was happening in this CAR T world. So I guess we'll do that, and then as we go along, we all know who are in practice this wonderful advance to do the CAR T therapy, and maybe if you occurs to you as we go through these abstracts, say a bit about their technology and what's new or different, as well as what the abstracts meant. So the first one, abstract 700, and it is entitled Primary Analysis of ZUMA-5, a phase 2 study of, we'll abbreviate it, Axi-cel, that product, in patients with relapsed refractory indolent non-Hodgkin's lymphoma. I think Carol Jacobson, the presenter. So what did you think this was the take home from this abstract?
Sure. Well, yes. So first of all, thanks for having me on. This is a fun forum, and one of the great things about being one of the ASH co-chairs is you do a deep dive into areas that you may not otherwise be diving as deeply in. So I'm a pediatric oncologist, as you know, and stem cell transplanter, and all of the studies that we're going to talk about today are focused on adult cancers. Now, as a sort of cellular therapist, and as most pediatric oncologists, I'm fairly agnostic about what diseases I look at, because the fields often advance in adults first and then move to kids. And so we can really, we like to keep our eyes on what's going on in the adult cancers to think about how we can apply them to kids. Now, the field of CD19 directed CAR T cell therapy is actually one of the very few examples where a new technology was actually rolled out in children before it was rolled out in adults. So this is kind of interesting. Yes, and in fact, it happened at the University of Pennsylvania and at Children's Hospital of Philadelphia, and so the CD19 CAR T technology, which was developed sort of simultaneously by several labs, but really the person who really did the most was Carl June at the University of Pennsylvania. And I know Carl well, and he's, just a real sort of hero in the field. And he developed the CAR T construct that is now the Novartis product. We're going to talk about that in a little bit. What you have here in the Axi-cel product was developed by Kite and is now owned by Gilead. Now, but the important sort of similarity between this product and the one that was first developed for pediatric leukemia, now has a lot of other uses, is that they all target CD19, right? So these are this sort of canonical B cell antigen. And what is notable about the ZUMA-5 study is that this is looking at the ability of what I'll call Axi-cel, because the full name is a mouthful. To treat patients that have not those acute lymphomas, but more indolent lymphomas. And so, as the title of the abstract suggests, they're looking at relapsing refractory, more indolent non-Hodgkin's lymphomas. And the 2 lymphomas that this study looked to develop the CAR T therapy in are marginal zone lymphomas and follicular lymphomas. And what's important here is not only the efficacy, but also the safety, right? Because when you have a more indolent lymphoma, you've got to be careful that your therapy isn't going to cause acute toxicities that would decrease somebody's survival. And so one of the really, I think, important things with this study was that the response rate in these quite difficult to treat more indolent lymphomas was quite striking. So if you took the follicular lymphomas where they had most of the patients, they had a 94% overall response rate, right? So that's
Very high.
really amazing with 80% CR. So really quite exciting. And then with the marginal zone lymphomas where, there are fewer patients and also is historically a harder to treat lymphoma, they still had a really high overall response rate. This was 85% overall response rate, 60% CRs and 25% PRs. So taken together, what this says now is that there is another treatment opportunity for these very difficult to treat patients. So that's fabulous. One of the sort of the cautionary tales I would say from this study was there was a grade 5 CRS. There was actually two bad cytokine release syndrome or CRS events in this study, one grade 4 and one grade 5 in one patient each. Now, that is not terrible when you have an acute, leukemia where you really are fighting against time. The occurrence of even one grade 5 CRS event is just something to keep in mind, obviously. So that means out of a total of 146 patients that were evaluable for those adverse events, there was one patient who died. So again, that's actually quite encouraging. But because safety is one of the most important things that we need to think about as we develop these therapies, it's just something to keep our eyes on. And I know that from speaking with the people who ran this trial, trying to understand sort of mitigators of CRS, and there's a big field in, that's focusing on ways to mitigate CRS. That will be important. And also trying to understand if there's a combination of patient attributes and cellular therapy attributes that might increase or decrease the risk of CRS.
So grade 4 or 5, bad enough, but as we go through these, because maybe the product's a little different or the patient population a little different, grade 1, 2, grade 3, everybody has something. I presume the cytokine release syndrome, the CRS, is happening everywhere.
Yeah, so if you look, for instance, in this study, 82% had any grade CRS. And what's really interesting here is, and this is for adult cancer, again, I'm a pediatrician. We practice in a different way in terms of cellular therapy where it's mostly inpatient. For adult cancer therapy, it is so important to get these cellular therapies in the outpatient realm because there's just not enough inpatient beds to care for everybody, inpatient who needs a cellular therapy. And also the idea, one of the ideas in the field is to make them, create treatment paradigms that are safe enough that you can move out of the tertiary and quaternary hospitals to more of the community hospitals where a lot of people are treated. And so to do that, grade 4 and 5 CRS is bad, but you really have to think about the grade 3 as well, because those less resourced hospitals aren't going to be able to treat patients adequately, even for grade 3.
Well, maybe as we go through them, we have 3 more to cover, but pause a
Mhm.
second, just if you could review a bit what's involved. So I refer a patient to the Zuma-5 study, and he or she has the requisite relapse refractory non-Hodgkin's, indolent, or marginal zone. What happens if you could just step through the process of collecting the cells, collecting the tumor, the T cells? How does the patient get involved, and where does this go? I know it's centered around.
Yeah. It's a really fascinating sort of cottage industry that has to come together for the patient in each one of these settings, because this is an autologous product, right? So a patient is enrolled on study, they're deemed eligible. They, do some basic testing to make sure their lymphocyte count is high enough to give a pretty good assurance that they'll be able to create a product for that patient. The patient then undergoes an apheresis procedure, so large volume blood, white blood cell collection procedure. That apheresis is shipped to the company. The company then does the transduction with the CAR construct, which is now really reduced to practice in many different ways. And there's really a very robust sort of system that has been created in multiple companies for creation of these patient-specific cellular products, really high transduction rates, really good quality control. I'll say that most of the companies still give the practicing physician back a sort of black box that says, here's your product, but we don't get the same sort of specifications that we get from other treatments we give patients. And I'm part of a working group nationally that's really working with the drug companies to try to improve our understanding of exactly what the transduced product contains in terms of the detailed cellular phenotype, right? We know that we get back the transduced cells, but still there's a lot of sort of black box in terms of which subtypes we're getting back for our patients.
So if we think of it then that my patient's army is collected by apheresis of patient's T cells sent to the company. Now, the patient's tumor is not sent, but the patient's army of T cells is sent. The
That's
company
right.
takes a lymphoma-specific construct to make that army then look for a lymphoma when infused back into the patient. Do I have it?
That's exactly right. So when I say that they've transduced the T cells, they've transduced to express what we call a CAR, which is a chimeric antigen receptor, which is able to see this, in this case, the CD19 molecule that's on all the T cells. So a T cell now recognizes this tumor antigen, CD19. And so in terms of examining the tumor, what the doctor has to do is to say, the treating physician, not the company, says, yes, this tumor does express CD19. The company then gives back T cells that can see that CD19 and activate the patient's own T cells that have been transduced with a CAR construct to kill those tumor cells.
Any problem with, so the bulk of what you have of the CD19 cells is your tumor, but you have other CD19 cells. Do we have too much toxicity there sometimes?
Well, that's considered an acceptable toxicity, but all of our normal B cells express CD19. And so for most patients, at least for a while after they get the CAR T cells, they also get, those CAR T cells not only fight the tumor, but they also get rid of your normal B cells. And so in some situations, you'll have to be given replacement immunoglobulin, for instance, to compensate for that. Yeah, exactly.
Okay. All right. I'm starting to understand it. All right. Let's then try abstract next, 1149.
Sure. Okay.
This one is efficacy and safety of, tisagenlecleucel in adult, close, in adult patients with relapsed refractory follicular. And now interim analysis of the phase 2 ELARA trial, Nathan Fowler. So what did you think of this study?
Yeah, I thought this was a great study. And I think the combination of these two studies really tells you where we are in the field. So this year at ASH, we didn't just have one company showing efficacy results for a CD19 CAR against indolent lymphomas. We had two. And in fact, this talk is the only poster that I actually included in my Best of ASH. And I thought it was a really notable study. I thought it had a lot of the same and really interesting points as the ZUMA-5 study did, and which is why I wanted to include it. I think it would probably be fair to say, again, this isn't my total area of expertise, but that these are competing trials. And I think that as a practicing physician, we want to know what is the breadth of our ability to treat the patients. And I would say that based on these two studies, it's probably fair to say that treating physicians either now or soon will have a choice of CAR T cell constructs from different companies. So this is the Novartis product, and it differs from the Kite or Gilead product in the signaling domain. So in one of the details of how the CAR construct is created. So both of these cellular products will recognize, again, that CD19 antigen that's both on the tumor and on normal B cells. They have a slightly different signaling domain. This Novartis product, it tends to be a little bit more long-lived, sort of more of a slow burn than the Kite product, which has a signaling domain that causes rapid expansion and then contraction. And I thought that the reason I included this was that I thought that that they were very similar in their outcomes. Again, there was no marginal zone lymphomas included in this study. It was all follicular lymphoma, very similar patient populations. And again, you saw very similar results. Overall response rate was 82%. with 65% CRs and 17% PRs.
Same
So
toxicity side effect problem?
in this case, there were no cases of grade 3 or higher CRS, and that may be due to the
Really?
different signaling. So, and again, this is, this is where the fine tuning of our choices may lie. So it may be that those 4,1-BB containing constructs are a little safer in terms of their early toxicity. But what we really need to know then is what the duration of the response is to really determine whether one will be better than the other. And really, it's just too soon to know. Like, for instance, this study, they reported out to 12 months. So that's not quite long enough yet.
So two similar patient populations, different products, and somewhat different results in terms of side effect. Okay.
Yeah.
So then the third one, abstract 544, entitled the Transcend CLL-004, a phase 1 cohort of liscabetagene maraleucel, or abbreviated Lysocel, in combination with ibrutinib for patients, again, relapse refractory. Here it's CLL, relapse refractory CLL, small lymphocytic lymphoma, and our presenter was William Weirda. So a different population, how did they do?
So yeah, so I thought this was a really interesting study as well. So now, again, this is a third. One of the things I also wanted to do in my talk is kind of highlight how many different companies are producing products for these sort of second line patient populations. So this is making sure I'm right. Lysocel is what people call it, and this is a Bristol Myers Squibb product. So again, sort of that's one of the themes, is that there are lots of choices these days. And this is a really interesting study because this is looking at patients with CLL. And what I thought was most striking about this study was it was combining the cellular therapy with the targeted therapy ibrutinib. And what smaller studies had identified was that by combining these two agents, so one targets BTK and the other targets the CD19, which are both important in CLL, that you could increase your efficacy and also your safety, because what the ibrutinib does is also it serves also to sort of calm down that sort of immune response. And what they found was that was exactly the case, is that that there were no grade 5 AEs or adverse events on this study. There were no grade 4 CRS or neurotoxicity events. So really quite safe. And when you looked at the all patients, all comers, the overall response rate was 95% overall response rate, with 63% CRs and 32% PRs. So again, very difficult to treat disease, CLL, and what I thought was most striking here was the successful combination with ibrutinib.
And I would suspect most of these patients probably had seen a BTK inhibitor.
Exactly. Yeah, that was so interesting, because it didn't matter if you had seen it before, you still responded when you added Lysocel to this. So I thought this was very interesting.
So it may have helped response and tapped down the AE.
That's right.
Fascinating.
That's right. Yeah.
Okay. All right. So our fourth one is abstract 556, entitled CD58 aberrations limit durable responses to CD19 CAR in large B-cell lymphoma patients treated with axibitagene ciloleucel, that can be overcome through novel CAR engineering. So even reading it is difficult. What did this show? This really got into the details of the science, sounds like.
Yeah, I have to say, as first of all, somebody who spends most of my time in the lab, this was my favorite abstract, I think, of the whole meeting. And in our sort of, with relevance to our conversation, it brings us full circle back to the first CAR T we talked about, which is the axi-cel, but with its first line therapeutic target, which was large B-cell lymphoma. So everything we've talked about on this call has all been dealing with CD19 CARs, but they're used with different diseases and some different CAR constructs. But what I loved about this abstract, it was really looking deeply at why a CAR T would fail, right? So when these CAR Ts first came out, we were all just extremely struck by their success. We were taking the highest risk patients, these patients that essentially had no chance for survival with conventional agents, and getting them into CRs. It was really nothing short of miraculous. But what we know now is that the long-term outcomes with CAR T cells are less than we would have hoped at the beginning. So while almost all patients, let's say even over 90% of patients will achieve a CR within 30 days of these products, the long-term duration of response is about 50%. So clearly there's a lot more that we need to do. So what this group did was really cool. They tried to identify whether there were any tumor specific factors. And so this gets back to what you were asking me at the beginning of our conversation. Can we look at the tumor to figure out why a CAR T may or may not work? And what they found was that in the patients that did not respond to the CAR T cells, the axi-cel, many of them had mutations in this molecule called CD58. So remember, this is a mutation on the tumor. And what we know about CD58 is it's very important in what we would call ligating this other molecule on T cells called CD2. So CD2 is a really important molecule for T cell engagement. And what it suggests is that even when you give these patients back these sort of magic cells, right, that have the CAR T in them, they still rely on some intrinsic T cell signaling to do their work. And in this case, if you give a patient a CAR T cell that has a CD2 that a normal T cell will express CD2, and some of these CAR T cells do as well. If the CD2 can't see CD58 on the tumor, the CAR T cell doesn't work. And so first of all, they figured that out, and that's a big discovery in itself. But then they did some engineering, and what they were able to do was create a second construct, so a second transgene, that they created a CAR T cell that not only contained the CAR, but also contained this hybrid gene that expressed that CD2 in a way that doesn't require the CD58. So they basically
got around that.
They got around it. And in mouse models, this thing cures tumors like gangbusters. And so what I thought was so cool about the study is they went to the patients, they figured out what, mechanism of failure, which is such an important thing in clinical investigations. Well, why do things fail, right? They figured out a molecular reason. They figured out how to engineer a T cell to overcome that. And then they showed, at least in an animal model, that could lead to clearance of a resistant tumor. And I'm sure that we'll be seeing this in the clinic sometime soon.
So I take this as why do CARs fail? So the tumors get smart and something may not be expressed or something may not be bound, ligands missing. And so this was a very nice science attempt and result on how to get around that and still show the CAR, the tumor, and have the desired result.
Exactly.
Okay.
Exactly.
Well, really no wonder you enjoyed the session, really, for interesting abstracts, taking it right on back to science. And yes, our Dr. June in Philadelphia is extremely popular and well known for all this groundbreaking work. So I guess lastly, where do you think this is going? There was probably some nice discussions and questions to you at the end of the session. This is great. What next? So I guess it needs to proliferate and get out of all the major university centers. That's one. And any new science out of the meeting or this session that you heard that might make it more available or help tap down the cost?
Yeah, I mean, the cost is really a critical barrier, and especially if CAR Ts themselves are not the end therapy for a patient. So for instance, as a transplanter, what we're wrestling with right now is how best to treat those relapsed patients. Now, this is not with the lymphomas that we've been discussing on this call, but with their first indication, leukemias. Should we use CAR T cells as a precursor for transplant for everybody, or can they be a standalone therapy? And I think the next generation of research is going to, first of all, determine that, but also to create CAR products that could be more relied upon to be that sort of final cure for either the leukemia and lymphoma. And if that happens, then you can better kind of swallow the high price tag. But I think we still need to develop those next generation CAR T cells that will be that ultimate cure for the patients that get treated with them.
Well, certainly fascinating technology, and I should have mentioned this last abstract on CD58 alterations was presented by Dr. Robbie Majner.
Mhm.
So I really want to thank you very much, Leslie. To remind our listeners, we've been speaking with Dr. Leslie Kean, Director of Pediatric Stem Cell Transplant, Boston Children's Hospital at the Dana-Farber Cancer Institute. Thanks so much. Really fascinating stuff, and it's only going to get better. We'll just stay tuned. Thanks so much for providing your insights today.
Thanks for having me.
Blood and Cancer is produced by MD Edge. Our editor is Jen Smith. All MD Edge podcasts are produced by Executive Editor Kathy Skarbek, and I'm your host, Dr. David Henry.